All right, deep inside our cells, there's this amazing molecular factory that literally builds fat from the ground up. It's called the fatty acid synthase complex. And trust me, if you're a med student, you're going to want to know how this machine works. So let's dive right.
But before we get into the nitty-gritty, let's take a step back and ask a really basic question. Why fat? Why did our bodies evolve to use fat as our main long-term fuel tank and not something like carbs? Well, it all comes down to one thing: efficiency. Just look at these numbers. Fat, or triacylglycerols, gives you 9 calories per gram. That's more than double what you get from glycogen. But here's the real kicker: water. Fat is stored completely dry. It's anhydrous. But every single gram of glycogen drags along 2 grams of water with it. And that, as you can imagine, has huge implications for how much we weigh.
And this is where it gets absolutely mind-blowing. Let's put that into perspective. If a 70-kilogram person had to store all their energy as glycogen instead of fat, they'd be carrying an extra 55 kilograms. That's an extra 121 pounds. It's just staggering, and it perfectly explains why evolution picks fat. It's the ultimate lightweight, high-octane fuel tank. Okay, so we know why we store fat, but how does the body actually build this incredible fuel so efficiently? Well, this is where we meet the hero of our story, a molecular assembly line known as the fatty acid synthase complex. We'll just call it FAS for short. So what is this FAS complex, really? The best way to think about it is like a giant, super talented protein that acts like a tiny factory inside our cells. You'll find it humming away in the cytosol, that's kind of the main fluid part of the cell, and it's especially busy in places like the liver, our fat tissue, and in lactating mammary glands. And its one big job: to build a 16-carbon fatty acid called palmate from scratch. Now, let's talk about how this factory is built. The FAS complex is actually a dimer. That just means it's made of 2 identical parts, or monomers, that are linked together head to tail. And here's what's so cool. Each one of these monomers has everything it needs: 7 different enzyme tools, plus one really important component called the acyl carrier protein. The complex only works when these 2 parts are together. And when they are, they can actually build 2 fatty acids at the exact same time. Super efficient, right? Okay, 7 different enzyme activities. I know what you're thinking. How on earth am I supposed to remember all of those for an exam? Don't worry, I've got you covered. There's a great mnemonic for this, and it's perfect for revision. Just remember this sentence: a cow can do really excellent baking. Okay, let's break this down. The A is for acetyltransferase. That's what loads the very first building block. Now we're going to jump to the end of the mnemonic for a sec. The baking part actually stands for malonyl-transferase, which loads all the other building blocks. A little out of order, but it works. Then we've got the C in cow, the D in do, and R in really. These are the core processing steps: condensation, dehydration, and the reductions. And finally, the E in excellent stands for the thioesterase, which releases the finished fatty acid. It's a bit of a quirky mnemonic, but it really helps nail down the different workstations in the factory. So you've got all these different tools, all these workstations, but how does the actual product, the growing fatty acid, get from one station to the next? Well, that's where the acyl carrier protein, or ACP, comes in. This thing is brilliant. It's basically a long, flexible robotic arm. It grabs the intermediate molecule and literally swings it from one enzyme to the next, with perfect timing and precision. This ACP arm is absolutely central to making the whole assembly line work so smoothly. All right, we've met the machinery and the workers. So now, let's put on our hard hats, head inside the factory, and watch this assembly line build a molecule of palmitate, step by step. Everything kicks off with a loading phase. Think of it like getting the first piece onto the conveyor belt. First, a 2-carbon starter unit from Acetyl-CoA gets loaded onto a specific holding spot on the enzyme. Then, the first real building block, a 3-carbon molecule called malonyl-CoA, gets attached to that robotic ACP arm we were just talking about. So now, we're all loaded up and ready to go. And now, the real action begins, a repeating 4-step cycle. It goes like this. First, condensation, where the 2 pieces are joined together. Second, a reduction, where NADPH comes in and donates some electrons. Third, dehydration. That's where a molecule of water is removed, creating a double bond. And finally, a second reduction, again using NADPH to get rid of that double bond. And boom, we've just added 2 fully saturated carbons to our growing chain. Now, this is the really important part to remember. That 4-step cycle we just walked through, its entire purpose is to add 2 carbons to the chain. So to get all the way from our starting piece to the final 16-carbon palmitate, that cycle has to run a total of 7 times. Each time a new malonyl-CoA piece is added to the assembly line, the cycle runs and the chain gets 2 carbons longer. It just keeps going until the job is done. So after 7 full turns of that cycle, we finally have it, a full-length 16-carbon chain still attached to that trusty ACP arm. This is where the last workstation, bioesterase, steps in. You can think of it as the quality control inspector who also has a pair of molecular scissors. It comes in, snips the bond, and releases the finished product, a brand new molecule of palmitate, off the assembly line. But, running a factory like this isn't free. It's actually a pretty big energy investment for the cell. Let's look at the price tag for just one molecule of palmitate. It costs 8 acetyl-CoAs, 7 ATPs, and a whopping 14 molecules of NADPH. That is a lot of energy and resources. And it makes perfect sense then why the body is so incredibly careful about when it turns this factory on and off. So, with that high energy cost in mind, the body obviously needs some strict controls in place. It can't just have this factory running 24/7. So the big question is, how does it regulate it? How does it decide when to make fat and when to stop? Well, the main on-off switch is hormonal. It's pretty straightforward, actually. When you eat a big meal, especially one full of carbs, your body releases insulin. Insulin is basically the signal for, we have plenty of energy, let's store some for later. It flips the factory switch to on. On the flip side, when you're fasting, your body releases other hormones like glucagon and epinephrine. These are the we're low on energy, shut down all non-essential production signals. They flip the switch to off. Simple as that. Okay, so we've gone deep into the biochemistry, but this isn't just about passing an exam. This stuff has major real-world clinical implications. So let's talk about what happens when this perfectly balanced system gets thrown out of whack. And the connections are huge. For one, if the FAS factory goes into overdrive, you get too much fat. This can lead directly to obesity and fatty liver disease. But here's something really fascinating. It turns out many cancer cells are addicted to fat synthesis. They hijack the FAS system and crank it up to the max to get the building blocks they need for their own rapid growth. This, of course, makes FAS a really hot target for new anti-cancer drugs. And on a different note, if you're deficient in vitamin B5, pantothenic acid, that mobile ACP arm can't be built correctly, and the whole assembly line just grinds to a halt. Okay, before we wrap up, let's just quickly recap those 4 core reactions. This summary table is a fantastic little tool for revision. It lays out everything nice and clearly. The reaction type, which cofactor you need, remember NADPH for those 2 reduction steps, and the specific enzyme that does the job. A perfect little cheat sheet for studying. And this all leads us to a really powerful final thought. Learning about this molecular factory isn't just an academic exercise, right? When you see how central it is to huge problems like obesity and cancer, you have to ask, could targeting this one complex, this one factory, be the key to a whole new wave of treatments? It's a really exciting possibility, and it all starts with understanding the fundamental biochemistry we've talked about today. Thanks so much for joining me.